Women's University has been known as a leader in quality education. In 1934, PWU moved into its main campus on Taft Avenue, and since the 1970s has been co-educational. Located in the heart of Metro Manila, it is easily accessible by public transport and surrounded by affordable housing. PWU School of Nursing has been offering high-quality, student-centered, and innovative nursing programs for over 6 decades. PWU's graduate programs upgrade the qualifications of nursing professionals, allowing them to advance in their chosen fields in the Philippines and globally. In the allied health fields, PWU pioneered in programs such as pharmacy, which was established in 1926 to train students who want to succeed in any aspect of pharmacy services. The first women doctors polyclinic in the Philippines was set up in PWU in 1951. In 1986, PWU's Bachelor of Science in Nutrition and Dietetics became the first nutrition program to be accredited by the Philippine Association of Colleges and Universities. PWU was also the first educational institution in the Philippines to focus on the prevention of acute illnesses and chronic diseases through its Master of Science in Clinical Nutrition program taught jointly with the St. Luke's Medical Center. PWU School of Medical Technology offers a competency-based curriculum enhanced by intensive training in clinical laboratory procedures. Offering a Bachelor of Science in Medical Technology and a Master's program in Medical Technology and Health Systems Management, PWU also offers a certificate program in phlebotomy. In nursing and the allied health fields, PWU continues to play an essential role in healthcare education, producing graduates who possess the skills that make them competitive in the country and anywhere in the world.
Good afternoon, everyone. Welcome to the Medical Technology Thesis Writing and Research Forum 2020.
Doing a research study is difficult enough. Putting it into thesis writing is scholarly demanding. The tasks would be daunting to a student. Fortunately, we have today's event that would give us an idea of the kind of topics to pursue, how to execute experiments, write the manuscript correctly, and successfully defend our thesis.
To commence with this very useful webinar, let us pause for a moment of silence in prayer and to offer the moment for our brothers and sisters who have been affected by the series of typhoons.
Praise God from whom all blessings flow. Praise Him, all creatures here below. Praise Him above, ye heavenly hosts. from whom all blessings flow. Praise him, all creatures here below. Praise him above, ye heavenly hosts. Praise Father, Son, and Holy Ghost. Praise God from whom all blessings flow. Praise him, all creatures here below. Praise him above, ye heavenly hosts. Praise Father, Son, and Holy Ghost. Praise Father, Son, and Holy Ghost.
With a fervently beating heart, let us place our right hand on our chests and pay respects to our national flag. Ladies and gentlemen, the Philippine National Anthem.
Let us warmly welcome the Program Chair of the School of Medical Technology, Professor Sherwin V. Toriano, for the welcome remarks.
To the Philippine Women's University community, to our Honorable University President, Sir Marco Alfredo Benitez, our SVP for Academic Affairs and Chancellor, Dr. Selina C. Young, guests, deans and program chairs, medical technology students, ladies and gentlemen, a pleasant afternoon to all. A thesis has a specific structure and content. It has to be true to form. It is the manifestation of experimental and scholarly pursuits that our students are expected to fully satisfy for the completion of the academic program, may it be in the college or in the graduate school. A term spent studying the course may not be enough, that we need to revisit the guidelines or accomplish several studies in order for us to master the writing of the research manuscript. Research studies is an authentic output that reflects the totality of a student's realization, skills, understanding, and application of the concepts in a multitude of threads in the medical laboratory sciences. It should satisfy the institutional rules that a medical technologist should pursue a study which is 1. Unique, 2. It should be experimental, at least for MSMT, and 3. It should be in line with the medical laboratory technology field. High-impact researches and exemplary thesis writing skills is the way to be more competent in a scientific world moving swiftly towards diversity and advancement. We decided on this webinar because it fulfills the standards of our STAR initiative. STAR means S for skills and training, T for technological knowledge, A for academic excellence, and R for research and collaboration. And I am giving this endeavor the highest mark of 3 stars. We have invited speakers who are top-notch in their field of study. Today's webinar will further widen our awareness and help us refocus as we pursue our vocation in medical laboratory sciences. The School of Medical Technology warmly welcomes everyone. Again, thank you very much and good afternoon.
Thank you very much, sir. As medical technologists, research plays a significant role in our course of study. Scientific advances and findings are products of research which are contributive to the medical field and to society as a whole. The positive application of this knowledge may equip us to battle diseases and help alleviate human lives.
Medical technology students and guests, we are honored today with the virtual presence of the Philippine Women's University President for an inspirational message. Ladies and gentlemen, please welcome Mr. Marco Alfredo Benitez.
Dr. Felina Young, PWU Chancellor and SVPAA, Mr. Sherwin Toriano, Program Chair of the PWU School of Medical Technology, Dean Nini Lim, Dr. Hipolito Palcon, our distinguished speaker, and thesis presenters, our hardworking faculty, students, and everyone watching on our social media platforms, good afternoon. First of all, I hope that all of you are safe and well in the middle of this global pandemic and in the aftermath of some major weather disturbances, last being Typhoon Ulysses, which ravaged Metro Manila and surrounding provinces just 2 days ago. I congratulate the organizers of this webinar, our School of Medical Technology, for continuing to provide our students the opportunity to further their knowledge, particularly in the field of writing and completing research studies, and its importance to the field of medical technology. Probably never has there been a period in our lifetimes where the spotlight has shone so brightly on the field of science, health, medicine, research, and medical technology as it has now with the world facing this global health crisis. From the discovery of the SARS-CoV-2 virus that causes the COVID-19 disease last December, to studying the ways by which it is transmitted from person to person in various environments and through different media, its effect on the human body, as well as the various treatments and eventually the development of a safe and effective vaccine against this virus. Scientific research and analysis have played a critical role. Never too has this research process, data, analysis, and recommendations been so highly scrutinized because of the dire effect this pandemic has had on the economies and populations of the world. The lives of millions of people and a return to some form of normalcy rests on the development of a safe and effective vaccine in record time. While this is the most pressing example, the importance of research studies from its conceptualization, writing, and completion is its contribution to generalizable knowledge of a particular field, whether that be in medical technology, allied health sciences, humanities, culture, music, the arts. The fact that properly done research is grounded on a systematic investigation, testing, and evaluation, the knowledge that is developed is generally reliable and backed by data. This reliability, accuracy, and trustworthiness of the data becomes all the more critical when public health is concerned as it is today. As a university, we believe that the research process which contributes to the furtherance of knowledge is one of our major responsibilities to society, and the onus is on us to ensure that our students are properly taught how to conduct, write, and complete research studies. This afternoon's thesis writing and research forum will hopefully be able to impart to our students valuable lessons for their own research projects. Once again, congratulations in advance to our presenters, and I wish everyone a safe and informative afternoon. Thank you very much.
Thank you very much. A round of applause for our University President, Mr. Marco Alfredo Benitez.
With great pleasure, let us usher in our resource speaker who will discuss the fundamentals of thesis writing. Let me call on our program chair, Professor Sherwin Toriano, to do the honors.
Thank you very much. Our distinguished resource speaker is a graduate of the University of the Philippines, Diliman, where he completed a double master's degree. One, Master of Arts in Education, specializing in mathematics and science. And two, Master of Arts in Measurement and Evaluation. He also finished the Doctor of Philosophy in Sociology and Anthropology at UP Diliman with a dissertation entitled Ethnography of Rituals, and he was awarded Outstanding University Research way back in 1992. From then on, he held several academic administrative positions. Let us begin with the Philippine Women's University, then EDSA, Quezon City. He was the former dean in the tertiary department. He was also the dean in the graduate school. He was the head of the Peace Education for Asia and the Pacific, which is an affiliate of the United Nations Disarmament Education Program. He was also the officer in charge in PWU Quezon City and the concurrent dean of the college and graduate school. In the Philippine Women's University, Manila, he was the director of the School of Distance Learning in 2005 up to 2006. He was also the director in the Expanded Tertiary Education Equivalency and Accreditation Program, or ETAP, from 2006 up to 2013. He was also the director in Research and Development from 2006 up to 2011. His research involvement includes the following: supervising statistician and design consultant for the Association of Non-Formal Education of the Philippines. He was also the consultant and Creative Spirit Consultancy of the Ateneo de Manila University for Community Survey, Social Acceptability. He was also the consultant for Tracer Studies and Research Office of the University of Batangas. He is also the resource speaker for the National Workshop of Competency and OBE Learning Outcomes for CHED. He is the chair of the MMHRDC Research Capability Building from 2005 up to 2011. And currently he is the member of the UP Manila Research Ethics Board. His published works include modules in methods of research and statistics with computer application. He is also a writer and contributor in Lifeline for Health and Aesthetic Magazine and Peace Economics Journal of New York, as well as in the Philippine Educational Forum Faculty Journal, the Journal of Graduate School, and Research in Focus. He also wrote in Ethnography and Education Journal, which was published internationally by Routledge, Taylor, and Francis Group. The development model for online instruction, written by him, was chosen by ASAIL to represent the Philippines as the country paper during the International Conference in Hong Kong in 2007. He is also a lecturer and paper reader for various national and international conventions. To name a few: the National Conference on the Filipino Teachers of SMED in Diliman in 2001, with the theme Qualitative Research, Observation Techniques. He was also the lecturer for faculty development in Chulalongkorn University, Bangkok in Thailand, with a research design meta-analysis written by him in 2004. Also, he was the paper reader for Asia Pacific Educational Research Association International Conference in Hong Kong in November of 2006. This is just a few of his long page of accomplishments. To sum up everything, he has been teaching the methods of research and statistics with computer applications from 1988 up to the present. Our very distinguished and accomplished resource speaker, ladies and gentlemen, please welcome Dr. Chipolito P. Falcon. Yes, sir, you can unmute now, sir, before we begin.
Naka-mute si sir.
Okay. Good afternoon to everyone, the university officials of PWU, from the dearest university president, the vice president for academics, the deans, the program chair of MedTech, Erwin Toriano, members of the faculty, and all those in the Facebook live stream. Okay. Thank you very much for inviting me. I'm going to talk about format and style in thesis writing. So it's a thesis writing, but that's the format and style in thesis writing. Next. Okay. Now, before we start with writing, we should set the paper for the margin by 1 inch top and below, and then at the right side. And on the left, should be 1.5 inch, using the paper 8.5 by 11. Okay, and the font should be 12 using Arial. The research paper or thesis contains the following parts. We have the preliminary parts, which consists of the title page, the approval sheet, the acceptance sheet. These two are provided by the office. The abstract, table of contents, list of tables, and list of figures. All these should be written in italicized word. The second part, which is the body part. Next, please. The body part contains the different, so these are the parts of a thesis. Chapter 1, introduction, background of the study, which is optional in some theses. Statement of the problem, hypothesis, if any. Then significance of the study, scope and limitation of the study, and definition of terms. What do you mean? Chapter 2 contains the review of related literature and studies. Related literature and studies, now at PWU, this is now combined, so it is not a separate topic. Both literature and studies should be written in terms of topics. So the topics for review of literature and studies should be topical. Then this is followed by a synthesis, and then a theoretical and conceptual framework. Sometimes theoretical, if you have a theory, to use in the research, and then conceptual framework. Okay, chapter 3, the methodology, consists of the research design, the respondents, the participants, or specimen in case for the researcher doing laboratory or experimental design. Then the data gathering procedures, the instruments, data analysis, and statistical treatment of data. In chapter 4, you have the presentation, interpretation, and analysis of data. And the last chapter will be the summary of findings, conclusions, and recommendations. So we'll go into the detail. We go on with this one. Then at the last, we have the bibliographical entries, the appendix, informed consent, and the curriculum detail. Okay. Now we'll start with the title page. You can show now the title page next. The title page, okay, this is just an example, a sample. The title page should appear in all capital letters. Yeah, it's okay, Sherwin. Okay. Centered and typed on the 8 double space line from the top of the paper. Recommended maximum length. It is recommended that it should be 12 or 10 to 12 only. But in some cases, maybe more than 12. But it is recommended that important words should be 10 to 12 only. Then the author's name should be in capital letters and on the 13th double space from the top of the paper. The submission statement should begin on the 21st double space line from the top of the paper. Okay. It is recommended that the title should be specific and concise. As much as possible, avoid the title starting with a study of, an analysis of, an evaluation of. In most of the theses that we encountered, this really not allowed. A study of, an analysis of, and an evaluation of. Okay, so you can see here that the title is all in capital letters. The name of the author should be capital letters, but all the others, except the other submission, the submission statement should be in small letter only. Okay, let's go to the next. The first one, the previous one is title page. This one is the cover page, title page. Usually this is the page where the binder, when you bind, for example, the thesis, this one comes out. Okay, so the same rule in writing, capital letters for title, and then a thesis presented to the faculty committee, the graduate school, the Philippine Women's University, Manila, in partial fulfillment of the requirements for the degree, Master of Arts in Medical Technology or Health Science System Management. And the name of the author should be capital letter. The year should be March, for example, March 2020 should be capital, but don't write the date, just the month and the year separated by comma. Okay. Now next is the table of contents. The table of contents. As I've said, the thesis consists of the table of contents. The table of contents consists of two parts. Of course, the preliminary italicized, the page italicized, and it is numbered using Roman numeral but small letter. Okay, so and after that, the chapter. We have the chapter 1, the problem and its background, and so on. Okay, so you have the page, you have the problem. It should be single space, but between title, between topics, double space. Okay, so that will be the same until chapter under chapter 2, 3, 4, and 5. But use Arabic numeral for the page. So the preliminary should be Roman numeral. Okay. So let's go to the next. This is now followed. The table of contents followed by the next page will be the list of tables. The word list of tables should appear in capital letters. 2 spaces below the page heading. Type the word list of tables. The table titles in the list of tables should have only the first letter of the principal words capitalized. So if we look at the example next. So, okay, so you can see list of tables. You have the number because if you have several titles or tables, you can have number 1, number 2, and so on. This is just an example. You have the number 1, the title. The first word is types. So only letter T is capitalized. Types of projects supported by non-government organizations in Metro Manila. But you also capitalize for example, except for proper names or proper nouns. It should be in capital letter. The first letter of the word. Okay, so mean performance of workers and so on. Quezon City is also capital letter because it is a proper name. Okay, so list of figures, we have the same format as the list of tables. So you have the number and the title of the figure. So these titles of figures, graphs, charts, and other illustrative materials. Okay, so these are the figures. It should be the same as the list of tables. First word theoretical, letter T is capitalized, all the others small letter. Okay. Then afterwards, and after that, I think we will have the abstract. But the abstract will be written after when all, for example, when the thesis is already edited, already through, before you can write the abstract. But the abstract should be indented, it should be in paragraph, but it should be not too long. Summary, summarize, especially a little bit of introduction, the background, the statement of the problem, the methodology used, the findings and conclusions and recommendations. Okay. So it's chapter 1. Chapter should be small letter except letter C, the first letter of the word chapter 1. Then the problem and its background should be all in capital letters. Then introduction, these are the topics now under chapter 1. Should be small letter also, but big letter for the first letter of the first word, introduction. Okay. So the statement of the problem, because we have some format and style, so I'm just pointing out, for example, parts of the thesis which usually uh, have the format and the style. For the statement of the problem, it's always uh, it can be copy paste. For example, the problem studied must be shown. Uh, there are two parts of the statement of the problem: the major problem and the specific problem. So the first paragraph should be the problem. The study is all what? The study uh, describe the study, look into the effect, whatever it is. Then the second paragraph should be specifically it answered the following problem. So you can have 3 or more problems there. So as I have read some thesis, for example, for the statement of the problem, let's start with introduction. So you already have your introduction, so there is no need of writing an introduction for the statement of the problem. Directly go to the major problem, then followed by specific problem. But they should be different paragraph. There should be indentation. Okay, we'll go to the next. Uh, for the hypothesis, we see that hypothesis is a conjectural statement of relationship between two or more variables. We have two parts, the null or the alternate. Usually, instead of writing hypothesis, you write the null hypothesis because null hypothesis is the one being tested. But it's also good to write the alternative or alternate hypothesis because if you reject the null hypothesis, then you accept the alternate, which becomes your conclusion. Was okay. So it's always stated in there are no significant. So it differs now. There are no significant what? Significant effect, significant relationship. There are no significant differences in means of and so on. Okay. Then we'll go to the conceptual framework. For the health systems management, usually if they go into correlative studies, it's better to make use of the IVDV relationship. By the way, conceptual framework, it's a figure. So therefore, the title of the figure should be below the figure. While in the table, the title of the table should be on top. Okay. Okay, so this is another example, the IPO, which is very popular to students. But for medical students, I I recommend that because it is experimental, you have to focus on conceptual framework using the design of the experiment. Whether you are doing pre-experimental design, through experimental or quasi-experimental design. So you have you can design it according to the you can design your conceptual framework according to given design of the experiment. Okay. But this one it can also be because your input will be your specimen, the process experimentation, and your output will be the result of your experiment. And then why do we have feedback? Because if our output is not so good, there are errors or mistakes or weak, we go back to the input and make it stronger, strengthen your input in order to have uh, a very good output. Okay, that's why we have that. That is the IPO, input process output uh, framework or the systems approach. Okay, now we will go to the presentation of data. Okay, so as an experience, most of the students really have difficulty in the presentation of data. The data are presented in graphical form, tabular form, textual form. In writing a thesis, it is always advisable to present a textual presentation, accompanied by a table or figure for a clearer understanding of the reader. By the way, when you write your textual presentation, the flow should go on. Just insert the table or the table or the figure. No, the figure is not inserted. The figure should be put in a separate page. It should not go with the text. The table can, if it's a small table. Okay. Now, writing the text. Writing should be double spaced. When quoting exact words or phrase, okay, exact words, so therefore there are quotation marks. Or if it is part of the paragraph, it is double spaced. Double space if it will not consume more than 3 lines. So when you write your quoted word, when you're writing, it's more than 3 lines, double space. So, so you'll go to the next, like this one. Okay. Sometimes we write double space, and then if, like for example, this one, the growing ratio between inanimate source of power, close quotation. Then you have the author, Levy, comma, 94, and then page 45. So the author, comma, the year of publication, colon, and then the page. No need of writing the letter P. That's understood. But usually, it's only the year and the page. But some writers use the author as part of the sentence, the text. For example, uh, Levy and mentioned, okay, or defined modernization as open quotation, the growing ratio between inanimate source of power, and you will have the year of publication and the page. But if it is more than 3 lines, you should indent. Okay, it should be written in single space, but it should be indented 4 steps again from the 7. When you indent 7, when you click the computer, 1, 2, 3, 4, 5, 6, 7, then you add 4 more to become 11. Then you start there. And then the next line will be in line with the indented 7 steps. Okay, so here you can put page 24 because it is alone. It is separated. It's not. It was not written after the author on top. Okay. So single space, more than 3 lines. If it is a word, a phrase, that less than 3 lines, it should be double space. Okay. When citations are made, use the author year on text citation. We call that author year on text citation. So there are the sample text gives you a complete pointers in citing authors for borrowed words or ideas. But when you cite an author where you just borrow the ideas, no more page. If there are no quotation marks, no page. But those are only in the year of publication. So when author is part of the sentence, author and then the year. Idea. When exact words or phrase are quoted, you have the text, the quotes, the author, year, and the page. Okay. Next. Follow the following steps in presenting the data for chapter 4. Write the topic based on the statement of the problems in chapter 1. Each problem is considered one topic for discussion. If it is in null form, for example, is there a significant relationship like that, where you have hypotheses? How will you write it? It should be written following the steps in hypothesis testing. State the null hypothesis, compute the statistics, present only the statistical table, state the level of significance, the degrees of freedom in the decision to reject or accept the null hypothesis. When you reject the null hypothesis, then alternate will be accepted. Therefore, you will say, finally, make your conclusion. You always say, I therefore conclude that there is, okay, a significant relationship. And then interpret and cite studies or literature. Some writers will cite literature or studies first before they present the data. Present first your data, make your conclusion, and then you can cite now literature, which will be literature or studies, if, for example, your result conforms with that or not, and so on. Okay. Now, tables. A table usually accompanies a textual presentation. This is presented in the text. Put on top or middle of the page. Don't put the table below or down because there is a tendency that the table will be cut, will be divided. So it's not good to look at below. It should be on top or at the middle. If you don't want to put it, for example, it's already below, so you can put it on the next page. When you put it to the next page, but your text is still in the previous page, mention the page of the table. But if the table is on the page where you're writing, no need of mentioning the page. Okay. If the table is complicated, so big, a bigger table, you need one page, solo flights, yeah. But it can be landscape, okay, horizontal direction. Okay. Figure, figures, a graph, a drawing, or a flow chart usually is put alone in one separate page. You don't mix this with your text. Mm. Don't mix it and make it very, very small. So it's not advisable. It should be alone in the separate page with the title written below the graph. So if it is a figure, it should be written below, center, below the figure. For a table, it should be on top. So this is an example of a table. It's put on top. Okay. Okay. Next, for the chapter 5, we have the summary findings. Usually, you summarize your a little bit of introduction, your methodology, the statement of the problem, the methodology, and then findings. Usually, we say findings based on the data presented to answer the statement of the problem. So, based on you can number your findings based on the number of problems you have. So, for conclusion, it is based on the findings. Make conclusions based on the findings. Mm. For the findings, there's no need of numbering it by 1, 2, 3, so on. You can write it in paragraph. Some writers will do that. Okay. So if you follow all these in writing, for example, and then the abstract, I think you have no difficulty, for example, to write your research for a referee journal. It's easier. Okay. Let's go to the next. Okay. Next page, please. Okay, we'll go to the bibliography. We follow the APA format, American Psychological Association format. So usually we have a book, for example, one author. So you just get the family name, comma, and then the first letter of the first name, or if there is a middle initial. Then in close and parentheses, the year of publication of the book. Okay, of course, the title should be italicized. All capitalize only the first letter of the first word of the title. Then if suppose you have two author book like Welter, comma, W, period, and Lloyd, comma, M.A. So you say Welton and Lloyd. You instead of writing A and D, you write the what do you call that? It is ampersand. Okay, in Lloyd. So Wilton W and Lloyd M.A. 2003. The title should be italicized also, capital letter for the first letter of the first word. Then, according to APA, if you have 6 or more authors in the book, you can just write the first author. So, for example, this one from Reads, comma L, then put et al. 2000. Okay, then the title italicized, and so on. Next, please. For articles, the author's last name, first name, initial, then in close and parentheses, date of publication, period, the article title, and periodical title. Periodical title should be italicized, comma, the volume number, then the issue, and then the page or the pages. For online documents, you should have the author's name, the date of publication, period. Title of work should be italicized. Retrieve month, day, year, and then from what? From a website, from full URL. Okay, others, then we will finish chapter 1, 2, 3, 4, 5. But by the way, when you put now the page, pagination, okay, the page should be right corner on top, except for chapter page. Chapter 1, 2, 3, 4, 5. The page for that should be down below, should be at the middle. You cannot see any book. You cannot see any book or any scientific, technically writing. The title of the next chapter, the page is at the right corner. Should be below. And all the pages, it should be right corner on top. Okay. While the preliminary parts, Roman numeral, the chapter 1 to 5 should be Arabic. Okay. Then you will have the appendix. We have the bibliography. Bibliography, you have already a list, and then you will have the appendix. These are the appendix. We call it Appendix A, Appendix B. It depends how many appendices you have. But some are saying that should be Appendix 1, 2, 3. We don't, because it was already given. Yung mga table, figures, and so on. So appendix is named Appendix A, Appendix B, Appendix C. Okay. Then another important document in research, I don't know if it is important for medical technologies because most of your researches don't make use of human subjects. So usually the animals and plants. But for human researchers, human participants, for the health system, you should have the informed consent. When your participants will be 12 years old and below, it should be signed by the parent. If it is 13 to 18, should be signed by the parent and the child. Okay. Adults alone, the adults themselves will. Okay, but in your informed consent, you introduce yourself, right? What is your purpose? Who are your participants, and what will be the participation of participants? And then how will you look into the risk and so on? And then below you should put the contact person and the contact number, so that if something happens, the participant will be able to call the researcher or anybody else. Okay, so another point is I have seen when you write in writing, the flow of sentences should go on. Okay, don't enumerate and then use bullets. You enumerate by using commas. Okay, so don't enumerate such as letter A, letter B, letter C, letter D. So don't do that. Okay, so I think that's all. Thank you very much, Sherwin. Thank you very much for listening.
Thank you, sir. Thank you.
Thank you very much, Dr. Hipolito Paragon. That was a detailed discussion on the proper contents and format of the thesis by which the medical technology students are required to finish. Again, let me call on Sir Kerwin to present the Certificate of Appreciation.
Wow.
Allow me to read the citation. The Philippine Women's University School of Medical Technology presents the Certificate of Appreciation to Dr. Hipolito P. Falcon as resource speaker for imparting his invaluable knowledge to a webinar entitled Medical Technology Thesis Writing and Research Forum 2020, conducted on the 14th day of November 2020, signed by Dr. Victor Emmanuel Gabriel, Forum Chair, and yours truly, the Program Chair. Again, thank you very much and congratulations, Dr. Hipolito Falcon.
Welcome. Thank you very much, Medical.
Once again, thank you, Dr. Hipolito Falcon. Medical technology experimental research studies are conducted in laboratories with the objectives of developing innovations, finding a solution, filling the gap in knowledge of answering scientific problems. Our Master of Science in Medical Technology graduates and professors were invited to present their studies, which were scholarly written as a thesis. Let us hear the first batch of presenters and after which open the floor for an open forum.
Our first thesis presenter, Dr. Ning F. Lim, has taught microbiology and clinical chemistry at PWU for nearly 4 decades. She developed a series of lecture and laboratory modules in bacteriology and mycology and began the PWU graduate degree program in Master of Science in Medical Technology and 1-month certificate course in phlebotomy. She received her Bachelor of Science degree in Medical Technology from the Philippine Women's University, completed her Master of Science degree in Biology at the University of Santo Tomas, and her Doctor of Education degree in Educational Management at the Philippine Women's University. She is also the former Dean and now Program Coordinator for the Medical Technology Graduate Studies. Once again, Dr. Nini Pastin Lim.
Okay, good afternoon, everyone. I would like to share with you the research findings that we had in collaboration with the UST Graduate School with my advisor, Dr. Maria Ruth Pineda Hortel. The title of the study is Plasma Ferritin in Gestational Diabetes Mellitus among a Filipino population. Gestational diabetes mellitus is a very important concern in obstetrics in medicine. GDM is a heterogeneous metabolic disorder, and it has an increasing prevalence. As a matter of fact, it has been observed to have 29% prevalence among Filipinos, and it is usually diagnosed on the 26th to 38th gestational weeks. Gestational diabetes mellitus could produce high blood glucose level in the mother, and this would bring extra glucose to the baby. And the fetus makes more insulin to handle the extra glucose. So why is it important to monitor GDM? GDM may alter the pregnancy outcomes, such as fetal and neonatal complications, maternal complications, and it may in the future affect the lives of the mother and the newborn. On the mother, it can cause preeclampsia, abortion, stillbirth, preterm birth, cesarean delivery. On the baby, it can cause fetal malformation, macrosomia or increased birth size, birth trauma, and neonatal hypoglycemia. On both the mother and the fetus, it can increase the risk of type 2 diabetes of about 40 to 60%. It can increase risk of prediabetes by 34.7%. It can increase the risk of obesity and hypertension. The objectives of the study were to compare the plasma ferritin levels between normal pregnant women with those having GDM and determine the possibility of using any as early biomarker of GDM. Specifically, it determined and compared serum concentrations of ferritin, CRP, and insulin on the first, second, and third trimester of pregnancy between GDM and non-GDM participants. And it determined and compared the age, BMI, hemoglobin A1C between GDM cases and non-GDM participants. The study was conducted at the University of Santo Tomas Graduate School Research Center upon the approval of the protocol by the Institutional Review Board. 90 pregnant women consented to participate in the study. Venous blood samples were collected. Blood serum were collected using gold top tube with gel separator for OGTT, glucose oxidase, lipid profile, enzymatic, and Friedwald methods, and the C-reactive protein using solid phase sandwich format immunometric assay. And lavender top with EDTA was used for the determination of hemoglobin A1C by boronate affinity assay, and the determination of insulin and ferritin was done using the sandwich ELISA. Statistical analysis was done using SPSS for the analysis, and mean and standard deviation was calculated to measure variables. For differences in continuous variables, t-tests was used. Spearman correlation was used for parameters, and a p-value of less than 0.05 was considered significant for all tests. The following were the analytical procedures done. Blood glucose was measured using the glucose oxidase method. The glucose using this method is determined after enzymatic oxidation in the presence of oxidase. The formed hydrogen peroxide reacts under catalysis of peroxidase with phenol and 4-amino antipyrine to a red quinonamine dye as indicator. Hemoglobin A1C was determined by boronate affinity assay using NicoCard. The kit contains test devices with a porous membrane filter, test tubes pre-filled with reagent and washing solution. The reagent contains agents that lyse red blood cells and precipitate hemoglobin, and blue boronic acid conjugate that binds with hemoglobin, an aliquot of which is added to the test device. Any excess of colored conjugate is removed with a washing solution, and the color intensity of total hemoglobin and the blue glycated hemoglobin is then read using a NicoCard reader. The Ray Bio human ferritin ELISA kits were used for the determination of ferritin and insulin. These kits are in vitro enzyme-linked immunosorbent assay for the quantitative measurement of human ferritin and insulin in the serum or plasma or cell culture supernatants. It uses an antibody specific for human ferritin or insulin coated on a 96 well plate. Standards and samples are pipetted into the wells, and ferritin present in the sample is bound to the wells by the immobilized antibody. The wells are washed, and biotinylated anti-human ferritin antibody is added. After washing away unbound biotinylated antibody, HRP conjugated streptavidin is pipetted to the wells. The wells are again washed, and a TMB substrate solution is added to the wells, and color develops in proportion to the amount of ferritin bound. The stop solution changes the color from blue to yellow, and the intensity of the color is measured at 450 nanometers. The RayBio human insulin ELISA kits is an in vitro enzyme-linked immunosorbent assay for the quantitative measurement of human insulin or proinsulin in serum plasma and cell culture supernatant. This assay has similar principle with ferritin assay, but it makes use of an antibody and biotinylated antibody specific to insulin. Nicocard C-reactive protein is a solid phase sandwich format immunometric assay. In the test well of the device, there is a membrane coated with immobilized CRP-specific monoclonal antibodies. When a diluted sample is applied to the test device, it flows through the membrane and the C-reactive proteins are captured by the antibodies. CRP trapped in the membrane then binds the gold antibody conjugate in a sandwich type reaction. Unbound conjugate is removed by the washing solution. The membrane appears red-brown with color intensity proportional to the CRP concentration in the sample. The following are the results and discussion. Comparison of means between non-GDM and GDM groups using profile. Findings of this study support the findings of other studies that age and BMI can be a significant risk factor in the development of GDM. Those who developed GDM were older as compared to the non-GDM group. The GDM group has higher body mass index, 27.003, which is considered overweight. Lau and Zhang found that obesity can be a possible cause of GDM, which is indicated by BMI. Glycated hemoglobin, or hemoglobin A1C, reflects the average blood glucose level within 3 months, the lifespan of the red blood cell. It has been used to diagnose diabetes and hyperglycemic control. In the study, the hemoglobin A1C of the GDM group is higher than the non-GDM group, but it is not statistically significant. Osmondson found that women who have hemoglobin A1C below 6.5 have increased risk for GDM, and some with with hemoglobin A1C below 5.7 develop GDM. Thus, it is not possible to use hemoglobin A1C alone in predicting GDM. The fasting glucose level in the GDM group was significantly higher than the non-GDM group, a high correlation with the GDM at a p-value less than 0.05. This supports the findings of other study that suggest an association of fasting glucose with GDM. Plasma insulin levels are consistently high on the first, second, and third trimesters in GDM cases, and significant at the second trimester, but not significant more than 0.05 on the first and third trimesters. The homeostatic model for insulin resistance, or HOMA-IR, is also high, more than 2. Insulin secretion is stimulated in the presence of insulin resistance because it was found that this is so that the body can compensate for decreased sensitivity. So you have a decreased sensitivity, increased resistance. Thus, increased insulin levels precedes the occurrence of type 2 diabetes. However, increased fasting insulin in the first trimester may only be observed in a particular group of cases. C-reactive protein is not significantly different between the two groups. C-reactive protein is increased in GDM, but is found to be affected by BMI. Thus, its usefulness as GDM predictor is unlikely. Combination of biomarkers tend to improve accuracy. How about ferritin? Higher ferritin levels have been found among GDM cases. Although ferritin is higher in GDM cases, there is no significant correlation has been found between ferritin levels and GDM cases. So not significant in the first, second, and third trimester. Ferritin is an important iron-containing protein. It is said to play a major role in iron metabolism. Its concentration in serum or plasma gives an approximation of the amount of iron stored in the body, or it can indicate inflammatory condition. There are conflicting studies, however, that showed whether serum ferritin plays independently as risk factor for diabetes mellitus, and if increased serum ferritin levels indicate iron stores or inflammatory conditions. So in conclusion, ferritin levels are higher among GDM cases, although still not enough to be used as early biomarker of GDM. Glucose is still the most important diagnostic marker for GDM. Maraming salamat po. Thank you very much. And terima kasih banyak. This is an Indonesian word. Thank you.
Thank you very much, Dr. Nini Lim, for that very informative thesis presentation. Moving on to our second thesis presenter, Ms. Canelli V. Canlas is a current instructor at the University of Santo Tomas, Department of Medical Technology. She was also a former faculty member at Our Lady of Fatima University, Valenzuela City, with subjects handled bacteriology, lecture and laboratory, mycology and virology, lecture and laboratory, and principles and strategies of health teaching in medical laboratory science. Lastly, she is a graduate of Master of Science in Medical Technology at the Philippine Women's University in 2019. Once again, ladies and gentlemen, Ms. Canelli P. Canlas.
Thank you so much for that introduction. Good afternoon. Once again, I'm Canelity Canlas. Good afternoon, Dean Nini. our Dean of School of Medical Technology, and also Sir Sherwin, Program Chair of the School of Medical Technology. And also good afternoon to the technical committee involved here in the research for made this possible. Once again, I'm Kaniliti Canlas. Without further ado, I will present to you the research paper that I have made last 2019, the activity of Leucidian Cagayanense leaf methanolic extract against MCF7 and angiogenesis. So here in vitro cell viability and the cytotoxicity assays with culture cells are widely used for cytotoxicity tests of chemicals and for the drug screening. So the application of these assays has been of increasing interest over recent years. And currently, these assays are also used in oncological researches to evaluate the both compound of toxicity. And because they are rapid, inexpensive, and do not require the use of animals. And furthermore, the human cancer-derived cell lines are fundamental models used in the laboratories to study the biology of the cancer and to test the therapeutic efficacy of the anti-cancer agents. And for the significance of this study, this research provided an important opportunity to advance the understanding of the different assays for the assessment of the cytotoxicity and as well as the anti-angiogenic property of my extract, which is the Glucidion cagayanense. Specifically, this study answers the following questions. Next. First, what is the effect of the Glucidion cagayanense leaf methanolic extract to MCF7 breast cancer cell line using the following concentrations? Here, from the protocol of University of the Philippines, Diliman, I have used the 5 concentrations, the 6.25, and you have here the 12.50 micrograms per ml, as well as 25 micrograms per ml, 50, and as well as the full concentration of the methanolic extract, which is the 100%. Next. And the second is there a significant difference in the effect of the Glucidion cagayanense leaf methanolic extract to MCF7, that is my cancer cell line, the breast cancer cell line, between each of its concentrations and the control. Next would be what is the effect of Glucidion cagayanense leaf methanolic extract to angiogenesis as regards with the length of the blood vessels and as well as the number of the branching points when used in the following concentrations. So here, I have used 2 assays in this research. The first, the MTT assay, that will check the percent inhibition of my extract, as well as the CAM assay or the chorioallantoic membrane assay that assessing the branch of the blood vessel as well as the length of the blood vessel. So lastly, in answer to the question, is there a significant difference in the effect of the Glucidion cagayanense methanolic extract to angiogenesis as regards with the length of the blood vessels and the number of the branching points between its concentration and the control? Next slide, please. The conceptual framework shows here that the study has dependent and the independent variables. As you can see, the independent variables used here is the methanolic extract of the Glucidion cagayanense leaf. For the NTT assay, there are 5 concentrations. You have 6.25, the 12.5, 50, and also the 25 micrograms per ml, and the 100 micrograms per ml. And another independent variable would be the methanolic extract of the Glucidion cagayanense leaf concentration. For the CAM assay, I have only 4 concentrations, the 25, the 50, the 75, and the 100%. And the independent variable will be the different concentrations, again, of the methanolic extract of my plant. For the methodology, the researcher used experimental design. The experimental design is a blueprint or procedure that enables the researcher to test the said hypothesis by reaching the valid conclusions about relationships between the dependent and the independent variables. Next. As you can see from the collection of the specimen, we are now in the methodology. The Glochidin cagayanense leaves, as you can see on the picture, the leaves were gathered at the Calao Cave, the Peña Blanca, Cagayan. Cagayan Valley is my mama's hometown, and it was carefully washed with the distilled water to remove the unwanted dirt. The next picture, okay, as you can see, the leaves were removed from the stem and the bark. And for the preparation and extraction of the plant, kindly return it to the last image. Return it to the last image for preparation and the extraction of the plant. As you can see a while ago, the picture that has been projected, no? From the identified sample, the leaves were sorted and air dried at room temperature, 20 to 22 degrees Celsius, for about 1 week or 7 days. And then the 1 kilogram of the dried marmarungay leaves were pulverized using a blender or the osterizer until I obtained the fine mesh produced. And I was able to soak it in 1 kilogram the 1 kilogram of the powdered marmarungay leaves in a 5 liters of the methyl alcohol for 48 hours. And as you can see on the previous pictures, no, I have used also the laboratory rotator, no, kasi needed to agitate the mixture, okay, to extract the constituents. And the mixture was filtered using the filter paper, and the filtrate was concentrated using the rotary evaporator for 50 degrees Celsius under the vacuum to extract the phytochemical constituents. And as you can see here, that is the duck egg that I have used for the CAM assay, and that will be explained on the next slides. Next slide. And this is the continuation of the research flow chart. It shows the chronological method to determine the cytotoxic activity of the plant, the marmarungay leaves, crude methanolic extract against the human breast cancer, which is the MCF7. And also, I assessed its anti-angiogenic property. Take note that I utilized the protocol from a journal for the preparation and experimentation to have an organized methodology. And for the NPT assay, back for the NPT assay, previous slide, previews, okay, the NPT assay or the NPT cytotoxic assay was performed in the study that was actually adopted from Mossman. Then again, that NPT assay was performed in the University of the Philippines, Diliman. Okay, in detail, no, the MCF7 are seated at the 96 well microtiter plates, and the plates are incubated overnight at 37 degrees Celsius and 5% carbon dioxide. And 5-fold of the sample uses treatments starting from the 100 micrograms per ml down to the 6.25 micrograms per ml. And the doxorubicin, as you can see, that is my positive control, and the negative control is the DMSO or the dimethyl sulfoxide. And after which, following the incubation, the cells were treated with each extract dilution. Again, I have 5 concentrations for this assay. And the treated cells were again incubated at 72 hours at 37 degrees Celsius and 5% of carbon dioxide. And then after incubation, the media were removed, and kaya siya tinawag na MTT because you will be adding an MTT, okay, an MTT dye at 5 milligrams per ml PBS. Okay? And then the cells, again, incubated at 37 degrees Celsius and 5% carbon dioxide for 4 hours. And after which, the DMSO, aside from being a negative control, it was actually used to dissolve the formaldehyde crystals formed by the reduction of the dye by the live cells. Next. And for the CAM assay, my second assay for this research, the one-day-old duck eggs were cleaned with 70% alcohol and grouped by treatments. So, ang ginamit ko dito, 10 replicates in each treatments, and then I have incubated at 37 degrees Celsius until the 7th day. And at that 7th day, we extract at least 2 ml of amniotic fluid. So I have extracted the albumin. Okay, tinatawag na albumin sa duck egg. That is the amniotic fluid. And I return it to the incubator. And on the 9th day, a sterile automatic pipette with the diluted extract was directly administered on the top of the chorioallantoic membrane assay. And the square window was sealed with a parafilm soaked in 70% alcohol. And then the egg was placed in an incubator again for 48 hours. So after 2 days na nag-incubate ko, the egg was open and the general condition of the cam was visually assessed. So I took a pic using a macro lens, and the picture was assessed using an angioquant, that is the software used to measure the branches of the blood vessel as well as yung length ng blood vessel was also assessed or measured using that software, the angioquant software. Next. For the result, I have here the percentage inhibition of the doxorubicin, that is my positive control, and the different concentration of the glucide cagayanense, the marmarungay leaves, the methanolic extract. The first table presents the percent inhibition of my plant extract against the cancer cell, the MCF7. So for the doxorubicin, as you can see, it gave a percent inhibition of 69.91. And as for the glucide cagayanense at 100 micrograms per ml, it's shown a percent inhibition of 67.58. And for the 50 micrograms per ml, it's shown 65.22. And for 25 micrograms per ml, it's shown 57.51% inhibition. And lastly, for the 12.50 and the 6.25, they shown a percent inhibition of 25.64 and 24.97 respectively. It shows here that the glucide cagayanense extract concentration increases, the percent inhibition also increases. And in addition to that, the percent inhibition of the 100 micrograms per ml of the plant extract is directly proportional to the percent inhibition of the positive control, which is the doxorubicin. And the second table, as you can see here, this is the comparison and the effects to the MCF7 between the glucide cagayanense, methanolic extract, and the control. The 6.25 micrograms per ml of the plant extract and the doxorubicin obtain a t-value of negative 36.31, followed by the 12.50 micrograms per ml of extract, and the doxorubicin obtained the t-value of negative 22.57. So the 25 micrograms per ml of the extract and the doxorubicin obtain the t-value of negative 11.17 and the t-value of negative 4.49 for the 50 micrograms per ml. It showed that there is no significant difference between the 100 micrograms per ml of Glochidium cagayanense leaf extract and the doxorubicin. Next. Okay, let's make it this quick. As you can see on the left picture, that is the untreated MCF7 cells, show that they have the intact membrane still. Okay, as you can see, the pseudopodia were at full stretch. Okay, that is the first picture, the untreated. And for that second picture, the left side, that is the MCF7 treated with 100 micrograms per ml of the marmarungay extract concentration. And on the right side, the positive control, as you can see, there is a complete loss of the cell membrane and it indicates a shrunken of the cells. So as you can see, there is no significant difference based on that picture. The concentration of 100 micrograms per ml of my extract and as well as the positive control, which is the doxorubicin. Next slide. So this would be the effect of the, let's project the table muna. The concentration, you have 25, the 50, the 75, and 100% of the concentration. Doon muna tayo sa table, sir. So the mean total number of branching points of blood vessels decrease as the concentration increase. Okay, that would be the summary. And as you can see, for the comparison and the effects of the branching points between the Glochidium cagayanense methanolic extract and the control, it reveals the analysis comparing the t-value of the total length of the blood vessels between the treatments. And as shown, as you can see, the positive control had significant difference between the 25% and the 50%, followed by the 75% and the 100% of plant concentration has no significant difference with my positive control here in the CAM assay, which is the retinoic acid. Okay, so 75% and 100% of the plant extract concentration, there is no significant difference with the retinoic acid, which is the positive control in the CAM assay. Okay, next. So those are the images captured after 2 days of incubation with my extract. And this, the comparison in the effects to the length between the Glochidium cagayanense, same with the branching points, the 75% extract of my leaf as well as the 100%, there is no significant difference with the positive control, the retinoic acid. Next slide. So the summary of findings, the concentrations are 6.25, and I use the highest concentration of the doxorubicin. The plant extract in 100 micrograms per ml has no significant difference with the positive control, which is the doxorubicin. While on the CAM assay, the methanolic leaf extract of the Glochidium cagayanense is successful in reducing the growth of blood vessels and has anti-angiogenic property, as evidenced by the reduction of the number of the branching points and the length of the blood vessels. Both the use of the retinoic acid and the Glochidium cagayanense methanolic leaf extract with 100% and the 75% concentration obtains statistically similar anti-angiogenic effects since there were no significant difference between the plant extract 100% and 75 and the retinoic acid, which is the positive control in the duck embryo using the CAM assay. Next. For the conclusion, the results reveal that the plant at the concentration of 100 compared to positive control shows non-significant. Apparently, the glucosidin cagayanense at the concentration of 6.25, 12.5, 50 compared to positive control shows significant result. This might be due to the heterogeneity of the phytochemical constituents of the glucosidin cagayanense and the sensitivity of the cancer cell line. Next. The conclusion, the the researcher was able to evaluate also the anti-angiogenic activity of the glucosidin cagayanense, the marmarungay, using the CAM assay. And based on the data gathered, the statistical analysis showed that 100% and 75 had no significant difference. Okay? And for the recommendation, further studies, the next slide would be the recommendations. Further studies use of other parts of the said plant to be tested for its cytotoxicity with the different cell lines, and as well as the isolation of a specific compound found in the plant and other parts of the plant for better analysis in determining its cytotoxic effect. And for the extraction solvent, to recommend from the non-polar to polar subjection of the marmarungay extract. And further studies should be conducted using other parameters and the inhibition of the angiogenesis and utilizing other image analysis aside from the AngioQuant software with a high resolution. And consulting an expert veterinary pathologist may be done to confirm results of the image analysis. And exploring the other plant species with anti-angiogenic properties would be another area for further study. And lastly, conducting a similar study with a plant species from the family Euphorbiaceae and in vivo cytotoxic activity of the glucosidin cagayanense kasi we have used the MPT assay and that is in vitro to evaluate more on how it will react in vivo system. I think that would be the last slide. Thank you for listening. If you have any questions later in the forum, no, you may write your questions and clarifications with regards to the presentation. Thank you for listening and have a good day.
Thank you, Ms. Canelli Canlas, for that excellent presentation. For our third thesis presenter, I introduce Ms. Shirley Joy P. Isip. She is a clinical instructor and faculty assistant at Our Lady of Fatima University. She won the first place during the oral research presentation in the Philippine Association of Schools of Medical Technology and Public Health and Philippine Society of Medical Technology Students National Student Congress. She also graduated from the Philippine Women's University in Masters of Science in Medical Technology in 2019. Ladies and gentlemen, our third research presenter, Ms. Shirley Joy P. Isip.
Thank you for that introduction. Once again, good afternoon to each and everyone. And I'm Shirley Joy P. Isip, RMT, MSMT, and I'm here today to present my research entitled Vaccinia myrtoides as histologic stain. So blueberries are classified in the section Cyanococcus within the genus Vaccinium. So Vaccinium myrtoides with a local name of Ayusip is a wild berry that grows in the mountainous and upland areas of Benguet province in the Cordillera region of the northern Philippines. So the blueberries contain a unique plant compound called the anthocyanin that provides their blue and purple pigmentation. The Vaccinium myrtoides is proven to possess anthocyanin content upon extraction using ethanol as its solvent. So this study was the first to evaluate its potential as histologic stain, particularly as a nuclear stain alternative to hematoxylin. So this research is beneficial for every medical technologist
for them to use as a potential organic, safe, and cost-effective way of staining the tissue samples in the histopathology section. So next would be the background of the study and the statement of the problem. So the main purpose of the study was to determine the staining ability of Vaccinia myrtoides ethanolic fruit extract as a histologic stain by specifically verifying the staining properties as regard to the pH, concentration, and the stability of the extract. It aims to answer the following questions. What are the properties of the extracts as regards to the pH and stability? What is the effect of the extract to the morphology of the appendix tissue when used in different concentrations? So I use 4 different concentrations from 25% to 100% of the extract. And lastly, is the staining effect of the extract to the morphological characteristics of the appendix tissue significantly related to its storage period? Next slide, please. So next would be the conceptual framework. As you can see on the slide, the input shows the resources and the materials that were used on the experiment. So we used blueberry fruit from Benguet, particularly Vaccinia myrtoides or IUCIP, and ethanol as the solvent, aluminum potassium sulfate as the mordant, and appendix tissue for the samples. The process includes the methodology of the research, and the output would be the final product, which is the histologic stain used as a nuclear stain for the tissues. Next slide, please. So for the methodology, 600 grams of fresh Vaccinia myrtoides fruit from Benguet was extracted with 2,400 ml of 95% ethanol. So the extract was subjected to a rotary evaporator to remove the ethanol solvent from the mixture. Next is to come up with the 4 different concentrations of the extract. The dye was diluted using distilled water as the solvent. So addition of the mordant, which is the aluminum potassium sulfate, it is mixed with the dye solution with a 1:10 ratio. So after mixing, the solution was heated for 2 minutes and cooled down for 30 minutes. So the experiment used appendix tissues. Okay, the tissue slides were prepared and stained using the routine histopathologic techniques that we are using in the laboratory. So the stained slides were read and graded by the pathologists. So to test the stability of the extract, the first set of tissues were stained with the freshly prepared staining concentration. And the succeeding sets of the tissue were stained on day 3, day 6, day 9 of the extracts that were stored on both room temperature and refrigerator temperature. So evaluation of the stability of the stain was done by 4 blinded pathologists by rating the groups of the tissue of the slides. So the evaluation of the stain was done with the guidance of a rubric depending on the observable tissue characteristics seen under the microscope. So the pH of the solution was also determined before the use of each extract. So next slide, please. So as you can see on the slide, these are the rubrics used for the research. So first would be the rubrics for the color effect of the extract. And we are looking for the following details. So we are looking for the uniformity of the color, if the slides or if the tissue samples are stained evenly. And then the next parameter would be the color saturation. So it will tell us if the color is intense or not, if the color is weak, if the color is fair, medium, strong, and vivid. So the next rubric is for the morphological characteristic of the appendix tissue. So the details that we are looking in here are the morphological characteristic of the nucleus and the cytoplasm of the tissues. And then we are also looking for the intensity again of the color. And then
the contrast and the clarity. So in here, we are looking if the nucleus and the cytoplasm are well differentiated to one another. So we are grading the staining effect by looking if the morphological characteristics are stained poor, sufficient, satisfactory, good, and excellent. So next slide, please. So properties of Vaccinia myrtoides ethanolic fruit extract. So these are the results of my experimentation. So the pH of the extract was determined using the pH meter. So the result of the pH implies that the extract was able to impart a color to the appendix tissue sample regardless of the pH and the storage duration. Although there is a slight increase in the pH after 3 days of the storage up to the 9 days of the storage, regardless of the temperature, the pH was maintained in its acidic nature and still can be used in staining the tissue samples. So based on other studies, the pH had a great influence on the stability of the anthocyanin pigment. So increase in the pH causes greater destruction to anthocyanin in samples. So anthocyanin were stable under low pH values and unstable under alkaline conditions. Also shown on the slide are the color representation of the appendix tissues rated by the pathologist. So as you can see, the medium color shows not too light and not too dark staining. The strong color contains moderately saturated and dark color. And lastly, the vivid color shows highly saturated, bright, and intense color. Next slide, please. So next would be the results on the morphological characteristics of the appendix tissue. So staining effects of the extract stored at room temperature to the morphological characteristic of the appendix tissue. So based on the mean rating of the pathologist, it can be observed that the 4 concentrations of the extract stored in the room temperature were able to impart a satisfactory to good staining effect to the morphological characteristics of the tissue throughout the storage period. On the other hand, staining effects of the extract stored at refrigerator temperature, it can be observed that the 4 concentrations of the extract stored in the refrigerator temperature had different staining effects to the morphological characteristics of the tissue from the fresh state to the day 9 storage. So for the results, the 75 and the 100% concentrations were able to impart satisfactory to excellent staining effect. The 50% concentration was able to impart good to excellent effect, and the 25% concentration was able to impart good staining effect throughout the storage period. It can also be noted that as the storage period increases, the staining effect of the extract stored at the refrigerator temperature to the morphological characteristics of the tissue also intensifies. Next slide, please. So as you can see on the slides, these are the representation of the appendix tissues that were rated by the pathologist using the rubrics or being guided by the rubrics in different concentrations and at storage periods. So as you can see, you can view the appendix tissue stained in the fresh state of the extract, appendix tissue stained in the day 3 storage of the extract, appendix tissue stained in day 6 storage of the extract, and lastly, the appendix tissue stained in the day 9 storage of the extract with different concentrations. So next slide, please. Next would be the relationship between the staining effect and the storage period of the extract. For the room temperature, there's no relationship in the staining effect and the storage period. So the staining effect of the extract stays the same over the period of storage in room temperature. On the other hand, for the refrigerator temperature, as the storage duration increases, the staining effect also intensifies. So it can be presumed that refrigerator temperature is preferred than room temperature as far as the storage is concerned. So according to other studies, temperature is another major environmental factor influencing the anthocyanin metabolism. So high temperature reduce anthocyanin accumulation in plants by inhibiting the expression of anthocyanin activator, and on the other hand, the low temperature induce anthocyanin accumulation. So next slide please. So as the conclusion of this study, Vaccinium myrtoides ethanolic fruit extract is a natural dye, can serve as a nuclear stain in staining the histologic tissue sample, or it can be used as an alternative with hematoxylin, which is commonly used inside the laboratory. The extract can impart a strong color to the tissue at the same time preserving the morphology of the cell. So we can view the nucleus and the cytoplasm of the cells easily. So based on the mean values among the concentrations evaluated by 4 blinded pathologists using a rubric, the 50% Vaccinium myrtoides ethanolic fruit extract stored at refrigerator temperature is the histological staining reagent that could be formulated in this study. So next slide, please. For further recommendations, future researchers can test for the viability of the stain. It can be tested for more storage days. Try other mordant aside from potassium aluminum or potassium alum, and give attention to the concentration and proportion with regards to the staining solutions. Other natural dye alternatives to hematoxylin as nuclear stain and eosin as cytoplasmic stain can be sought. Different tissue types can be used in order to determine the efficacy over all types of tissues. Future researchers can also use the extracted dye in blood samples, other body fluids, or even microbiological specimen. So other factors that affect the anthocyanin can be set as parameters for the study, such as the light, oxygen, and the solvent. So next slide, please. So before we end, I would like to take this opportunity to thank the Philippine Women's University School of Medical Technology, Sir Sherwin Toriano, Dean Minilim, and I would also like to extend my regards to my research advisor, which is Ma'am Felicitas Aquino. Okay, so there's a reason why we don't see the world in black and white. So once again, thank you for listening. God bless and have a good day.
Thank you for that enlightening and extensive research discussion, Ms. Shirlene Isip. We will now open the forum for the question and clarifications. May we call on Sir Sherwin Toriano and Ms. Felina de Guzman for the open forum part 1.
Okay, thank you very much. For our open forum, we have here the first question. And the first two questions goes to Dr. Lim. Dr. Lim,
Yes.
the question goes, when should ferritin be tested in pregnancy to be an aid in the diagnosis of GBM? And the second one is aside from your Ray Bio human ferritin ELISA test kit, is there any alternative assay that can be used for the study?
Okay. For the first question, why ferritin is studied?
For
Yes,
pregnancy,
yes, because in many studies, although they have been conflicting, no, and most of these studies recommend further studies for ferritin as a biomarker for GDM. So this study would like to see whether it can be used as a an early biomarker for GDM. Actually, this research is not yet complete because the targeted participants really is 200. But we only had that time, last November 2019, we only had 90 participants, which we have to follow um, strictly from first, second, and third trimester. Actually, we had many participants, but one limitation of the study, one difficulty that we encountered is that not so many of them, would come back and had their second trimester and third trimester examination. So we have to extend so that we can, we will be able to have the targeted number of patients. Since we have to present it already in Indonesia, we tried to analyze the 90 pregnant women who participated in the study. So the tests are still being continued until this time. I hope we will be able to finish everything by December. By December, we had a delay because of the pandemic, and no one is allowed to use the laboratory facilities at UST during the pandemic. No? So there is a delay, but I think this time we'll be able to continue with the tests, and probably by December, everything will be finished. And I think by the time we will really be able to make strong conclusions and make recommendations with regards to the use of serum ferritin, insulin, and other biomarkers for GDM. Now for the second question, ano nga yung second question?
There's another alternative for your ferritin
For the Ray Bio. We tried to make a canvas of the ELISA kits. And I think this is the one that more or less could be afforded, no? Because we have to do 2 trials or duplicates, and one plate will only have 96 wells. So that would mean 48 samples only can be used. And you can just imagine the number of kits that we need to use for the 90 and even for the 200 targeted participants. Now, among the kits, the one that is most available and I think most affordable is the Ray Bio. So we concentrated on Ray Bio, and the Ray Bio was very cooperative. They responded to our request right away for the kits. And I would like to thank my advisor who really helped me in all of this. No? And she is Dr. Maria Ruth Pineda Cortel of UST. And of course, I would like to thank the whole GDM team of Dr. Cortel, who helped me in the collection of the specimens, because to be able to follow up the patients, you really have to stand by the hospital and be there once the patients would come and have their blood extraction.
Yes, Ms. Kulina, I think we have a second question going to Ms. Canla.
Yes, our next question is for Ms. Canlas. Ms. Canlas.
Yes.
Our question, our question for you is, what is ingloculion that is cytotoxic to cancer cells?
I have
Thank you.
quantitate the phytochemical constituents of the plant. Okay, it reveals that it contains flavonoids that contributed a lot with the cytotoxic effect of the said plant, the flavonoids, as well as the saponins, the triterpenoids, and the alkaloids. Also, those are the phytochemical constituents contributed to its cytotoxicity as well as the anti-angiogenic property.
Thank you, Ms. Canlas. Our last question is directed to Ms. Isip. Ms. Isip,
Yes.
what is the effect? What is the effect of pH and storage on the staining capability of the extract? Thank you.
So as I discussed a while ago, the pH contributes on the staining intensity of the extracts. If or it is also associated with the stability of the anthocyanin. Okay, so in my results or based on other studies, the pH had a great influence in the and they are stable in the acidic pH, they can able to impart color on the tissue. So, sa madaling salita, dumidikit yung mga parts or yung stain on the tissues based on the pH. And what is the other question? Aside from pH, what is the
Storage.
For the storage naman, we tested if yung stain ba talaga is gumagana pa siya or it is effective kahit na matagal mo nang store yung ating the stain itself. So for the storage, the relationship is that as we store the stain, particularly on the refrigerator temperature, is mas nag-intensify yung stain niya. So you can relate it to the ripening, kung familiar yung mga medtech dito, ripening of the stain. So as you can associate it to the hematoxylin. Okay? So that would be all.
Thank you, Ms. Isip. Let us proceed with the second and last batch of our thesis presenters. Our fourth presenter is none other than Mr. Sherwin B. Toriano. He is a graduate of Master of Science in Medical Technology in PWU School of Medical Technology 2018. He commissioned in microscopic researches for intestinal parasites in microfilaria. He is also a trainer and a resource speaker in parasitology. Moreover, he also founded the Philippine Society of Laboratory Microscopists, which is an educational organization dedicated to learning parasitic and cellular elements using its social media platform. Lastly, he is the current program chair of School of Medical Technology in Philippine Women's University. Ladies and gentlemen, our fourth presenter, Professor Sherwin Toriano.
Hello, good afternoon. My thesis is about the differentiation of blastocysts from insect vectors and its pathogenicity to Swiss mice. Blastocystis species is a protozoan parasite transmitted by the fecal-oral route by human-to-human or animal-to-human transmission. Its role as a pathogen is still debatable, but current studies have tilted the balance where it is gaining more acceptance as causing gastrointestinal symptoms. Insect vectors are known to carry disease-causing microbes. However, do they harbor blastocystis? Only a few studies have been made on the isolation of parasites from insect vectors, and none have studied the pathogenicity of these isolates on mice. The inadequate knowledge about these prevalent parasites The controversies surrounding the nature of its infectivity and pathogenicity, including the ambiguous findings in its detection using routine smear microscopy, are very compelling reasons to direct the research efforts on this. Hence, this research study was undertaken to answer the following. What is the differential count of blastocystis from insect vectors, namely Periplaneta americana, Musca domestica, and Drosophila melanogaster species using various staining media, including gram, acid fast, methylene blue, and Lugol's iodine stain? Is there any significant variation in the differential count of blastocystis from insect vectors when grouped according to the staining media used? What is the effect of staining media to the morphology of blastocystis? Is there any significant variation in the effect of staining media used to the morphology of blastocystis? And finally, what is the pathogenic effect of blastocystis isolated from insect vectors to our Swiss mice? My conceptual framework follows the systems approach. The input includes all the materials, including the isolated blastocystis from insect vectors, namely your Periplaneta americana or your giant cockroach, your Drosophila melanogaster or your fruit fly, and of course, your Musca domestica species or your common housefly. It also includes staining media such as the gram stain, the methylene blue, the acid fast, and of course, the Lugol's iodine used for your direct smear. And we are employing Swiss mice for the test of pathogenicity of the isolate. The process involves all the methods in the experimentation, such as the staining methods used in microscopy. Also, we included the purification of the parasite, which is required in order to facilitate the inoculation of our Swiss mice. And after the mice were inoculated, we studied by fecalysis the infectivity level on the feces of our infected mice. And finally, we checked for pathogenicity by doing histopathologic studies on the intestines of our Swiss mice. The output is the realized objectives of these theses, and they are the parasite differential count among the insect vectors, the comparative morphologic differentiation using the staining techniques, and it requires the alternative cultivation and the purification protocol used prior to the infection of the mice. And we also were able to study or to observe the feces of our induced mice. And finally, we checked for the histopathologic tissues for the pathogenicity of our blastocystis isolates coming from our insect vector on the intestines of our Swiss mice. For the research methodology, the research study is a three-part experiment involving isolation, differential count, morphologic studies, mice inoculation, and histopathologic studies about blastocystis. The insect vectors were determined by purposive sampling. They are randomly selected from all the specimens collected all over Metro Manila. The insect vectors included Periplaneta americana or your giant cockroach, your Drosophila melanogaster or your fruit fly, and of course your Musca domestica or your common housefly. The specimens were the external washings from these insects and the intestinal contents coming from these insects. For the microscopic techniques, we made use of staining media, such as the acid-fast staining, ground staining, methylene blue, and your Lugos iodine for your direct smear. The stained smears were microscopically viewed by 3 certified microscopists in order to determine the count of the blastocystis parasite per smear and the morphology of blastocystis according to their staining characteristics, which include the clarity, color reaction, cellular differentiation, and of course, the overall visual quality as rated by means of the rubric scale. The concentration and purification of our inoculum. All the tubes of the collected specimens per insect vectors were combined into one tube. These were the external washings, including your intestinal content. To render it suitable for inoculation, the blastocystis suspension was concentrated and purified in a medium which supports blastocystis growth, and it requires the Ringer's solution added with human serum. The inoculum was then standardized to contain 10,000 cysts per inoculum volume in order to infect each of the 10 mice in the group. The inoculum was given orally through a feeding tube attached to a tuberculin syringe. There was no isolated blastocystis from our fruit fly sample. Also, we added a negative control to compare the effects of the blastocystis containing inoculum with an inoculum which is just blank. For the mice experiments, 30 Swiss mice, 10 to 12 weeks old, were prepared. They were housed individually in partition cages to inhibit unnecessary contact with each other. The mice were fed in a normal diet of commercial pellets and given potable water ad libitum. The mice were maintained in an animal laboratory at 25 degrees centigrade under a 12-hour light and dark cycle. They were tested negative to blastocystis bificalysis prior to inoculation. Now, all the mice were monitored for the presence of infection to blastocystis daily for 10 days after they were inoculated, and the average count of blastocystis detected were then recorded. After 10 days post-inoculation, all the mice were dissected to remove the entire length of their intestines, which were submitted for testing. Tissue samples of the mice intestines were fixed in 10% formalin, embedded in paraffin, sectioned and stained in hematoxylin and eosin stain, and examined under the light microscope by a veterinary pathologist. The cellular changes in the mice intestines were observed for signs of pathogenicity. Now let us proceed to the findings. Table 1 is the differential count of blastocystis from insect vectors determined using various staining media, and this one answers the statement of the problem number 1. It is noteworthy to say that the parasite count in your Periplaneta intestinal contents were generally higher than the count in the external washings. However, the parasite count is quite similar in both specimens external washing, and intestinal contents in your musca species. Now, the studies of Tatang and L. Sherbini showed the presence of parasites in these insects as well. But these studies failed to note the presence of blastocystis in them. Take note also that we did not find Drosophila, blastocystis parasite from our Drosophila specimen. Drosophila melanogaster is your fruit fly. Now, in a study conducted by Zimmer in 2012, Schlenke et al. Conducted a study further, and it was known that yeast actually ferment the sugars in the fruits. And this created the alcohol concentration in these fruits, which are actually at a concentration greater than the concentration of alcohol in a bottle of beer. So the researchers observed that the Drosophila flies eat the yeast, including the alcohol that they produce. And so this might be the explanation why we did not find blastocystis from our fruit fly or Drosophila species. It is because the alcoholic content of the fruit made by the yeast is actually toxic to blastocystis. Next, in our findings, we have table number 2. Table number 2 is the variation in the differential count of blastocystis from insect vectors when grouped according to the staining media used. And table number 2 is an answer to the statement of the problem number 2. And there is no significant variation in the differential count of the parasites among the insect vectors across specimens using a 5% margin of error. Now, after the differential counts were obtained from the specimens, the evaluators or the microscopists were then tasked to grade the morphology of blastocystis according to their staining characteristics using your rubric scale. So as you see, the example is grade 1 for A. That is the visual acuity for grade 2. And an example for grade 3 in your rubric and for grade 4 in our rubric. And this staining characteristic using your rubric is used in our table 3. So table 3 shows you the effects of the staining media to the morphology of blastocystis. And this table is an answer to statement of the problem number 3. Now, considering the mean of the rubric findings in every staining medium, that they were higher actually in your methylene blue and direct smear as compared to your gram stain and acid fast stain. The mean in your gram stain and acid fast, according to the evaluation of our microscopist, is just 1.0. And 1.0 means that our microscopists were able to see visible the cell membrane and vaguely delineated the intracytoplasmic vacuum. However, when compared to using the methylene blue and the direct smear, the microscopists were able to distinctly see the cell membrane, and there were visible large central vacuoles and your peripheral nuclei. Now, the gram stain, when used to blastocystis, was also studied by Artita et al. In 2017. In the blastocystis stain, gram stain, the cytoplasm of our vacuolar forms took up the counter stain safranin. This is because they do not possess a peptidoglycan layer. In our A of B staining reaction, they do not possess a thick mucopolysaccharide layer, leaving them stainable with our counter stain. And for methylene blue, as your wet mount preparation, the peripheral cell nuclei, which contain the DNA, are usually identified microscopically as the darkest areas of the cell stain. So this stains the acidic structures of the parasite, which gives them partial differentiation. Table number 4 is the variation in the effects of staining media to the morphology of blastocystis from isolated from our insect vector. And this is in answer to the statement of the problem number 4. Now, the statistical evaluation of the morphologic findings of the isolated blastocystis when viewed microscopically with the various staining media showed no significant variation in both the intestinal contents and the external washing, except in Musca domestica external washing. And these may be due to the kind of cysts present or are isolated, which were more amenable to staining with the methylene blue than in others, which was also detected in the study of Al-Kubri in 2016. Lastly, I am showing you plate number 2, which is the composite pictures of the severity, showing the severity of blastocystis pathogenic effect in the intestines of the Swiss mice. So as you can see, these are the microvilli supposed to be found in the intestines of mice. And according to our study, majority of the infected mice showed villi erosions. 17 over 20 of them showed villi erosions from moderate to severe. The negative mice control, on the other hand, showed results from none to unequivocal mild to moderate villi erosions only. So there were no blastocystis found embedded in the tissue. There were no inflammation or no signs of lysis as well. Now, all of the findings leads us to these conclusions. In view of the findings presented, which point by point considered the statements of the problems, the researcher concluded that Periplaneta americana, or your large giant cockroach, and Musca domestica are carriers of blastocystis parasite in their intestines and even in their external appendages. Also, gram, acid-fast, direct smear, and methylene blue staining methods can be equally can equally detect the presence of and differentiate blastocystis from all the samples, as there was no significant difference in the differential count and the morphology of the parasite across the methods used. And finally, blastocystis species from isolated from our insect vectors were found to be infectious to Swiss mice, and the infection resulted to the erosion of the intestinal villi of the Swiss mice in both the small and the large intestine. Blastocystis infection is just luminal, meaning that it's just freely floating in the diameter of your intestine. Histologic sections showed no signs of tissue invasion, nor does it trigger inflammatory processes. So again, thank you very much and good afternoon to you all.
Thank you, Professor Sherwin Toriano, for that interesting research presentation. And last but not least, our last presenter is Mr. Clive Ivan Mercado, presently a molecular analyst at Eastern Visayas Regional COVID-19 Testing Center. He was a former full-time instructor, Department of Medical Technology at St. Scholastica's College, Tacloban, and part-time laboratory supervisor at the research department. He has also earned his degree of Master of Science in Medical Technology at PWU School of Medical Technology 2019. Once again, our last thesis presenter, Mr. Clive Ivan S. Mercado.
Thank you so much. Viruses are not just harbingers of death and suffering. Some of them are actually friends. Ladies and gentlemen, a pleasant afternoon. Happy National and Science Technology Week. And hello there, I'm Clive Ivan Mercado, your friendly neighborhood virus researcher, and welcome to your place of inspiration. Without further ado, there are two important words that can be found in the research title. Number one is Klebsiella, and number two are bacteriophages. Let's talk about the bacterium first. To begin with, Klebsiella is a genus of bacteria classified under family Enterobacteriaceae. The most important and perhaps the most dangerous species classified under this genus is Klebsiella pneumoniae. Klebsiella pneumoniae is a gram-negative lactose-fermenting non-motile capsulated bacterium that is strongly associated with septicemia, pneumonia, urinary tract infection, and even the globally emerging disease such as pyogenic liver abscess complicated with metastatic meningitis. Based on the 2013 Declaration of the U.S. Centers for Disease Control and Prevention, Klebsiella pneumoniae has been classified as one of the three groups of microorganisms with the highest threat level, meaning this group of bacteria are immediate public health concerns that require aggressive and urgent action. Today, there are already strains of Klebsiella pneumoniae that are resistant to nearly all antibiotics, including aminoglycosides, cephalosporins, fluoroquinolones, and carbapenems. This silent pandemic of multi-drug resistance, ladies and gentlemen, is felt even as far as here in Eastern Visayas because from 2015 to 2018, Klebsiella pneumoniae ranks as the second most common bacteria from clinical specimens capable of synthesizing extended spectrum beta-lactamase, or the enzyme that we fondly call as ESBL, capable of degrading penicillin type of antibiotics or the beta-lactam antibiotics, thus making the strains of bacteria multi-drug resistant. And from 2017 to 2019, it ranked first among clinical isolates in the Divine Word Hospital based on the research conducted by Lim et al. A bacteriophage, on the other hand, as seen on my research title, is a virus. Again, it is a virus, and a virus is not a living organism. For in fact, I am one of the few researchers who would like to believe that a virus is just a rather complicated molecule. Anyways, a virus on its own is a submicroscopic, obligate intracellular parasite. There are two types of viruses depending upon the host that they infect. Number one are the animal viruses, and number two are the bacterial viruses. All viruses that infect human beings, such as HIV, rabies, Marburg, Ebola, and the infamous coronaviruses, are all classified as animal viruses. Ladies and gentlemen, these are the enemies of life. Bacterial viruses, on the other hand, infect bacteria. Yes, you heard it right, a pathogen capable of infecting another pathogen. And unlike antibiotics, bacterial viruses or the bacteriophages can be compared to heat-seeking missiles that destroy their hosts' cells with surgical precision. In other words, they will only target a certain spectrum of disease-causing bacteria. Therefore, we can harness this potential to truly combat the silent pandemic of multi-drug resistance. This is precisely the reason why we conducted this study. Next slide, please. The objectives are the following. Number one, we need to isolate a lytic bacteriophage against ESBL positive Klebsiella pneumoniae divine word hospital strain. So why use this bacterium as the initial host? Well, because it's pathogenic and it's highly deadly, thus making it a scientifically reasonable initial host. And the second objective is to determine the host spectrum of the bacteriophage that was isolated from Mangon Bangon River. Now, so why include this as an objective? Well, The danger of bacteriophage therapy is the possibility that these viruses could infect bacteria from the indigenous microflora. Therefore, we need to find out the host spectrum of these viruses in order to ensure that they will only destroy disease-causing microorganisms. And the third objective is that we need to describe the morphology of the bacteriophage using transmission electron microscope. Well, as the classic saying goes, to see is to believe. Transmission electron microscope is the most powerful imaging tool in our generation used to visualize viruses. And if we can't observe the structure of these viruses under the electron microscope, it will be very difficult for us researchers to synthesize the primers that we need for polymerase chain reaction. The next objective is, next slide, is find out the initial identity of the bacteriophage using polymerase chain reaction. Since there are no commercial kits to identify bacteriophages at the molecular level, this is described as initial identification. We need to do a full genome sequencing in order to know its true identity. In the case of my study, I was not able to do it because of the cost. And the last objective is to determine the response of bacteriophage inoculation in rabbits using WBC differential count. A WBC differential count will enable us to observe the course of a controlled dose of bacterial infection and viral load in a given host. I use rabbits because they are better animal models compared to laboratory rats when you are running assays in order to satisfy the Cox postulate. And let us go now to the conceptual framework. Again, let us move to the conceptual framework. In order for us to be able to characterize bacteriophage from Mangon River, the following were done. So we did 4 assays. Number 1 is the double agar overlay plaque assay and the fudge spot testing. So this assay was done in order for us to determine the host spectrum of the bacteriophage that we were able to isolate. So we have here an array of bacteria. Most of them are gram-negative except for Staphylococcus aureus. So we have 2 strains of Klebsiella pneumoniae, 2 strains of Staphylococcus aureus, 2 strains of Pseudomonas, and then we have Salmonella enterica subspecies enterica, Salmonella enterica subspecies typhi, and then 2 strains of Escherichia coli, and well, we also included the infamous Acinetobacter baumannii in the host spectrum of bacteria that we have tested against the Klebsiella predating bacteriophage isolated from Mangon River. And then we also did transmission electron microscope in order to visualize the bacteriophage, and then polymerase chain reaction and in vivo analysis. So the expected output are the following. We have number 1, to describe the in vitro lytic activity of Klebsiella predating bacteriophages, and then describe its morphology based on the electron micrograph from the TAM, and then initial identification using marker genes through polymerase chain reaction and in vivo lytic activity of the Klebsiella predating bacteriophage. Now, let us go to the methodology. Again, the methodology. The research design used is descriptive analytical. Sampling technique was purposive sampling. Again, it's purposive sampling. And then we used this t-tests concerning 2 independent means and one-way analysis of variance for our statistical analysis. Now, let us go to the data gathering procedures. Again, as you can see on that slide, there is a picture that describes sterilization of glasswares and another picture that describes the collection of water samples. Now, glasswares were first autoclaved for 30 minutes. Sampling was done in between 9 in the morning to 11 in the morning of March to April 2017. Prior to this, trial runs were done in January and February 2017 to test the efficiency of the isolation techniques for the viruses of interest. These days were strategically chosen because they were predicted to provide a climate of partly sunny weather with room temperatures in between 31 to 33 degrees Celsius. Such condition prevented the dilution of the selected sites due to the rainwater and the formation of unnecessary algal overgrowth. Once this suspected water samples are incubated together with the host bacterium, and then We have here the preparation of Luria bertani agar and the preparation of host organism and the fudge lysate. Now, Luria bertani agar, again, fellow researchers who are interested with bacteriophage technology, Luria bertani agar is the primary plating medium for double agar overlay. Again, let me repeat it. Luria bertani agar is the primary plating medium utilized for bacteriophage plaque assay. As per the protocols established by the Cold Spring Harbor Laboratory, the formulation is presented on the slide. We have here casein, enzyme hydrolysate, yeast extract, sodium chloride, non-nutrient agar, and adjust it to final pH of 7.5. 2 milliliters of each collected water sample was usually added to 2 ml of TSB or tryptone soya broth. 100 microliters of the host bacterium, which in this case is Klebsiella pneumoniae ESBL positive, was then added and incubated at 37 degrees Celsius for 24 hours. Such setup was done in order to amplify the initial concentration of the bacteriophages by allowing them to feed on the host bacterium overnight. After the incubation, the test tube containing the suspected bacteriophage and the host bacteria was then centrifuged at 5,500 revolutions per minute for 5 minutes. The supernatant, take note, it was the supernatant and not the pellet that was aspirated with a sterile 5 ml syringe and passed through a 0.22 syringe base membrane filter. This is done in order to remove the host bacteria from the virus of interest. So therefore, what we have here in our supernatant is a very pure concentration of bacteriophages, and this serve as our stock fudge lysate. Now, next slide, please. We have here the double agar overlay method. Again, it's a double agar overlay method, and I will say it again. This is the primary assay that is done in order to determine the spectrum of bacteriophages' lytic activity. Bacteriophages were titrated using double agar overlay method. So you might be wondering, why is it called double agar overlay method? It's because of the fact that we are using two types of agar. Number one is Luria bertani agar. And again, this is the primary plating medium. It contains about 3% agar, and the function of Luria bertani agar is to provide minimum nutrients for our bacterial cell host. And the second one is the soft agar on top, which consists of only 0.7% non-nutrient agar. Again, we have here 0.7% non-nutrient agar. It's called soft agar because it's more liquid in consistency and it has very low nutrient concentration. So the reason behind this setup prevents the host cells in forming colonies. Thus, it also actually enables the researcher to observe bacteriophages when they start forming plaques. Now, we have to count and calculate titers and do fudge spot testing in order to properly describe the spectrum of bacteria that are infected by the bacteriophage that was isolated from Mangon-Bangon River. So again, we need to count and calculate titers and then do fudge spot testing. So how do we do this? We have to count plaques. Plaques are small, clear spots that form through the cloudy area over the plate where bacteria had grown. These plaques are patches of dead bacteria, and each of them represents one plaque forming unit. So what? The viral titer is a quantitative measurement of the biological activity of bacteriophages and usually expressed in plaque forming units or PFU per ml. In order to calculate for the titer, the plaques were taken out of the incubator and these plaques were counted. In other words, the plaque count is directly proportional to the bacteriophage lytic activity. Now, we did transmission electron microscopy and polymerase chain reaction as mentioned earlier. The bacteriophage suspension was obtained by centrifugating it a 5 ml mixture at 25,000 G for 60 minutes, followed by resuspension of the pellet in 100 microliters of SM buffer. Preparations of fudge particles were processed by standard protocols for positive staining and transmission electron microscope. In detail, 10 microliters of the concentrated fudge suspension were allowed to adsorb on a carbon-coated matrix and then stained with 2% uranyl acetate. That's the standard stain that we use in transmission electron microscopy. The grid was subsequently washed 2 times with double deionized distilled water, air dried, and imaged by TEM. Polymerase chain reaction, on the other hand, was carried out by two processes. First of all, we have to extract the genetic material of the bacteriophage, and then through purification using nucleus spin virus DNA and RNA isolation kit that was manufactured by Biorad. And then we have to do polymerase chain reaction. So as you can see here in the next slide, we have here genomic primers of selected Klebsiella predating bacteriophages. Again, these are genomic primers of selected Klebsiella predating bacteriophages. So you might be wondering, how was I able to what do you call this? How was I able to synthesize this? So we have to log in the National Centers for Biotechnology Information and use the application for Primer Blast so you can develop your own primers. So, and then we go now to the inoculation of host bacterium and bacteriophage and then plating of host bacteria from test animals in the next slide. Again, inoculation of host bacterium and bacteriophage and then plating of host bacterium from the test animal. Rabbits were infected intravenously with 0.5 ml of an ESBL Klebsiella pneumoniae strain. Usually, the concentration of the host bacterium is 5 times 10 raised to 6 CFU per ml. We are not using the standard inoculum because we need to inject a sublethal dose. When the symptoms appeared in 48 hours, animals were administered with treatment. So the first group received 0.85% normal saline, the second group received gentamicin, and the third group received a 0.5 ml bacteriophage suspension with a concentration of 1 times 10 raised to 8 PFU per ml. So the blood samples of these rabbits were extracted through femoral vein puncture and were placed in ethylene diamine tetraacetic acid tubes. So these were subjected to hematology analyzers, and another sample was taken in order to satisfy the Cox postulate. Now, let us go to the results. Again, let us go to the results. As you can see in this picture, you can see a healthy bacterial cell at the logarithmic phase. So these are healthy vegetative K pneumoniae cells undergoing binary fission as seen under the transmission electron microscope. Take note that the bacterium in this electron micrograph is free from bacteriophage infection. Now, let us look at the picture at the electron micrograph when bacterial cells are incubated with bacteriophage from Mangon River. So, electron micrograph of K pneumonia cells, as seen under this slide, they're already ruptured and dead. So this proves that the bacteriophage are lytic in nature. And then, as next slide, we can see here a slide about the host spectrum of the bacteriophage isolated from Mangon River. So, the bacteriophage isolated from Mangon River was able to lyse Klebsiella pneumoniae, ESBL negative, and ESBL positive, and then as well as E. coli, ESBL negative, and ESBL positive strains, and then at the same time Salmonella serotype typhi. It can also be observed that lysis of host cells was still seen even when the phage lysates were diluted at 10 raised to 6. So this is where the industrial significance of phage research comes in, because even if you dilute it 1/1,000,000, there is still lytic activity, or they are still destructive against the type of host bacteria that they tend to infect. And then let us now go to the structure of the bacteriophage, or the morphology of the bacteriophage as seen under the transmission electron microscope. Ladies and gentlemen, I am proud to show you the first ever electron micrograph of bacterial viruses here in Eastern Visayas. And I am proud to say that this was done by a research conducted in the Philippine Women's University. The morphology of the purified bacteriophage particles was examined using transmission electron microscope as described earlier. So since I am not a professional virus researcher, I don't have a degree on virology. These images were sent to the University of Granada in Spain for confirmation. So while we were waiting for confirmation, we initially named the bacteriophage KP-MBQ3, named after the host of the bacterium and the site from where it was collected. And then let us now go to the initial identification through chain reaction. Now, the initial identification of KP-MBQ3 or the bacteriophage that was isolated from Mangon River was determined through PCR amplification of the phage DNA using a genomic primer for KP34. KP34 is a bacteriophage that was already profiled in the NCBI or the National Centers for Biotechnology Information. So based on the type of host that it infects and the type of morphology it shows under the transmission electron microscope, this is the closest lead that we have to the true identity of KPMBQ3. So electrophoresis showed that there was an amplicon with an estimated size of 350 kilobase pair that migrated through the gel. So based on the results of the morphology and then the DNA amplification, it can be suggested that this bacteriophage belong under order Codovirals and family Podoviridae, again, based on its morphology under the transmission electron microscope and on the DNA amplification results, it can be classified under order Codovirals and family Podoviridae. Now, let us go to the whole spectrum of the bacteriophage isolated from Mangon River as seen during the in vivo studies. So it was observed that the rabbits artificially infected with a controlled dose of Klebsiella pneumoniae were cleared of infection after receiving an intravenous suspension of KP-MBQ3. This can be reflected by the absence of growth on McConkey agar and a sharp decrease, as you can see in the slide, in the number of granulocytes. So a sharp decrease in the number of granulocytes tell us that indeed there was a clearance of bacterial infection. And as you can see, there was a sharp rise of lymphocytes. So therefore, it could be that the bacterial proteins were released into the extracellular environment and and then our lymphocytes were reacting to them. Thus, there is an elevation in the result of the CBC. It was also further observed that during the re-isolation of the bacteriophage, not all remained viable after circulating for 24 hours within the peripheral blood of the artificially tested animals. So this remains to be a mystery even for me, the researcher, because we were not able to further this study from this point onwards. So I was not able to explain why some rabbits showed that there were no bacteriophages already circulating in the peripheral blood, so we were not able to further the study at that point. Now, for the conclusion, we were able to find indeed a bacteriophage lytic against an extended spectrum beta-lactamase producing strain of Klebsiella pneumoniae that was isolated from Mangon River. And in terms of its host specificity, it can lyse a narrow spectrum of gram-negative bacteria that most of them belong to family Enterobacteriaceae. And as seen under the transmission electron microscope, the bacteriophage closely resembles a podovirus. Next, electrophoretic separation showed an amplicon with an estimated size of 350 kilobase pairs, thus telling us that it could be, again, phage KP MBQ3 could be indeed bacteriophage KP34 as profiled in NCBI. And then number 5, rabbits artificially infected with a controlled dose of ESBL-positive Klebsiella pneumoniae were cleared of infection after receiving an intravenous suspension of KP-MBQ3. But again, we were not able to fully explain why there were rabbits showing no viable bacteriophages in their peripheral blood after circulation within 24 hours. And recommendations are the following. So we invite other researchers not to increase the repertoire of host bacteria that we are testing against our bacteriophage. So normally bacteriophage can be found in the environment, in soil, in water, in the sea, wherever there is high degree of fecal contamination. Most of the time you can see bacteriophages. So, but the thing here is that we need to understand more on the type of host cells, host bacterial cells that they infect. So based on the CDC recommendation in 2013, the following are recommended. So it's a very good starting ground for young aspiring researchers. So we have here Clostridioides difficile. We have here Carbapenem-resistant Enterobacteriaceae, Neisseria gonorrhoeae, Campylobacter, Salmonella and Shigella species, Streptococcus pneumoniae, and Mycobacterium tuberculosis. And at the same time, determine the antibiotic susceptibility of each host bacterium. So in my case, I was not able to do it because it was a bit expensive. No, when you test for antibiotic susceptibility, especially when you are using the Vitic system, may kamahalan po siya. But the quality of the results are very good. And lastly is to find out if there is an antagonistic or synergistic relationship between bacteriophage, lysate, and antibiotics. So based on the frequency of clinical use, the following are recommended for synergistic evaluation. We have here trimethoprim, colistin, ciprofloxacin, gentamicin, amikacin, meropenems, imipenems, cefepime, ceftriaxone, amoxicillin, ampicillin, cefixime, tazobactam, and clavulinic acid. So that's all. Thank you so much to my dear alma mater, the Philippine Women's University and the College of Medical Technology, through the leadership of Sir Sherwin Toriano and Dr. Ninilim, for giving me the opportunity to be able to present my research in this thesis and research forum. May the force be with you all, and science without God is madness. Again, may the force be with you all.
Thank you so much, Mr.
Thank
Mercado,
you,
for
Clive.
the
Thank you.
presentation. We will now again open the floor for the questions and clarifications. And at this juncture, I would like to call my co-MC, Ms. Belina de Guzman, to raise any questions from our viewers with regards to the presentation of Mr. Mercado and Mr. Toriano. Belina?
Okay. Thank you, Rania. Our first question is directed to Sir Toriano. Sir Toriano, what are your recommendations that would help people avoid misidentifying blastocystis as other parasites, especially in morphology characteristics? Thank you.
Okay, so blastocystis is a ubiquitous parasite, meaning that it can be seen all over. So most of our animals, insects, even humans may have blastocystis. It can, animals and humans can harbor the parasite without having signs and symptoms as well. So you would not really know unless you have been tested for blastocystis. Now, blastocystis in humans is called in most literatures as blastocystis cuminis. Blastocystis and then the word cuminis, meaning or is referring to humans. However, recent advances and literature tell us that blastocystis cuminis is now a misnomer because blastocystis found in animals can also be found in humans, and blastocystis found in humans can also be found in animals as well. Now, in identifying blastocystis, you really need to have training, okay? Years and years of training in order to study and to master the morphology of blastocystis, because blastocystis is a kind of parasite which is very unique. It has 4 forms. So unlike your amoeba, which has 2 forms, the cyst and the trophozoite, your blastocystis has 4 forms. So you can master 1, 2, or 3, but generally you cannot master all 4 because the 4th one, which is the trophozoite or the amoebic phase, is very elusive to find. Okay? However, you can find the elusive amoebic stage in diarrheic stool as well in humans, including animals. Now, in order for you to correctly identify blastocystis, you need to really train. And for those medical technologists who are actually working right now or who are licensed and are already practicing in clinical laboratories, miquas may be sent to them which may contain blastocystis. And to tell you frankly, There was one year wherein blastocystis was part of the nucleus, and I even misidentified that blastocystis from the specimen. So from then on, my experience of having misidentified this blastocystis compelled me to pursue a research study on blastocystis because I think I have been exemplary in the microscopy of all other parasites except in this blastocystis parasite, which gave me really too much difficulty when it comes to microscopy. And now my research studies about blastocystis. My faculty study is also about blastocystis. And even some of my qualitative researches are about blastocystis. And in order to study and to master blastocystis well, it has to be a purposive sampling. You go somewhere else, you collect any kind of sample that you can have, not just human feces, not just from insect vectors, but from anywhere. Okay? Again, you have to train in order to not to miss out on the proper identification of blastocystis.
Thank you, Sir Toriano, for that answer. May I call on my co-MC, Ms. Rani Maulana?
Thank you, Felida. So this question is directed to Mr. Clive Ivan Mercado. So you actually have two questions. So the first question is, if you were to make interventions and more variables, what would it be? The second question would be, can you tell us more about the macrophage naming? On how did you came up with that name? The part when you are waiting for the identification.
Okay, thank you for the rather well difficult questions. Number one, I would like to address the first question if I would rather make interventions. So first of all, I've already mentioned in the recommendations not to study or to increase the spectrum of host bacteria that you want to understand what as to which of this bacteria can your fudge lies. Okay, so those were recommendations by the Centers for Disease Control and Prevention. At the same time, I still also want to do a synergistic or antagonistic study between bacteriophages and commonly used antibiotics, wherein now these commonly used antibiotics are already being laughed upon by the bacteria that we are dealing with in the lab. And at the same time, if I were to conduct another research about bacteriophages, I would go to the great outdoors, look for more sources of bacteriophages because viruses are just literally everywhere. They are the most abundant source of genetic material in the biosphere. So if you want to research on viruses, then go to the great outdoors, go to the coral reefs, go to soils contaminated with fecal material. I guarantee you there are bacteriophages out there. And to address the second question, thank you, Ma'am Ami, for the rather difficult question. Yan, nakakatakot ang tanong niyo po. Actually, the naming was just agreed upon by me and some of the colleagues in the lab who helped me work with the bacteriophage. So actually, it was called KPMBQ3 in our lab. We call that virus KPMBQ3. KP, the first two letters stands for the host cell that it infects, the initial host cell, which is Klebsiella pneumoniae. And then MB stands for Mangon-Bangon River. And then Q3 stands for Quarry Area Number 3. It's an area in the Bangon River that we call as Quarry Area Number 3. So most of the time, usually when we do naming of viruses while we are waiting for their true identity, is that first two letters, bacterial host, second letters are the site of collection, and the last letters are if you want to specify it more. So yun, kaya siya KPMBQ3. But if you were to name viruses, again, if you were to name viruses, what I would advise is that you look for primers that are unique for these bacteriophages, preferably primers that target what do you call this, target reserved genes for these bacteriophages. Usually nakikita niyo po siya sa NCBI. So just see to it that once you visit NCBI, at the back of your mind, you have a prior knowledge. What bacteriophage are you dealing with? What's the possible suspect that I am handling with in the laboratory? So from that point on, you can use the primer blast application, and then makikita nyo po doon that it will try to synthesize primers based on the gene of interest that you want to amplify. So after that, kung may amplification po siya sa primer nyo, you can do genetic sequencing afterwards. Yun nga lang po, may kamahalan, but again, that's the best way to identify your bacteriophage. And at the same point in time, you can also prove that if the bacteriophage that you are dealing with is a novel organism. So I hope that gives justice to the questions that were thrown at me. Thank you so much. May the grace be with you.
Thank you very much for that interactive open forum. We hope that the research speakers have addressed all of your questions. To express our gratitude to the speakers, may we call on Professor Sherwin Teriano to award the Certificate of Appreciation.
The Philippine Women's University School of Medical Technology presents this Certificate of Appreciation to Dr. Nini Christine Lim, thesis presenter, for imparting her invaluable knowledge in the webinar Medical Technology Thesis Writing and Research Forum 2020, conducted on the 14th day of November 2020. Signed, Dr. Victor Emmanuel Gabriel, the forum chair, and yours truly. Congratulations and thank you very much, Dr. Lim.
Thank you. Thank you very much. Thank you.
Next, the Philippine Women's University School of Medical Technology presents this Certificate of Appreciation to Miss Sherlyn Joy P. Isip, thesis presenter, for imparting her invaluable knowledge in the webinar Medical Technology Thesis Writing and Research Forum 2020, conducted on the 14th day of November 2020. Signed by Dr. Victor Emmanuel Gabriel, forum chair, and yours truly. Next, the Philippine Women's University School of Medical Technology also presents this Certificate of Appreciation to Ms. Canily P. Canlas, thesis presenter, for imparting her invaluable knowledge in the webinar Medical Technology Thesis Writing and Research Forum 2020, conducted on the 14th day of November 2020. Signed by Dr. Victor Emmanuel Gabriel, forum chair, and yours truly. Next in line, the Philippine Women's University School of Medical Technology presents this Certificate of Appreciation to Mr. Clive Ivan S. Mercado, thesis presenter, for imparting his invaluable knowledge in the webinar Medical Technology Thesis Writing and Research Forum 2020, conducted on the 14th day of November 2020. Signed, Dr. Victor Emmanuel Gabriel, forum chair, and yours truly.
Thank you so much, Sir Sherwin.
To culminate the program, we are very honored to have with us the PWU Director for Graduate Studies. Ladies and gentlemen, please welcome Dr. Emerita Zornosa.
Thank you. Good afternoon, everyone. Once again, thank you to all of you, the PWU Medical Technologies community. It has been a pleasure being with you all today. We appreciate the engagement and all the exerted efforts and valuable contributions you put to this first virtual research writing forum 2020. We are so blessed today for having the valuable contributions and ideas and experiments and research results that is very informative and very helpful to the medical fields and medical care. Most especially to mention Dr. Hipolito Falcon, that we have been provided a very clear and complete guidelines on the formats and forms where the content should be placed in the correct position and its proper chapters. Expect this can help you reduce stress in doing your next research and future researches. We gathered several important insights on medical technology field as this forum requires. Thank you to Dr. Nini Lim for sharing her research on the improvement of maternal and newborn health, wherein ferritin level elevated has associated with type 2 diabetes. Thank you also to Ms. Carnelli Canlas for initiating an experiment regarding marmorungay leaves methanolic extract that can be a potential anti-angionetic sorry, properties that can lead to the development of medicine for treating cancer. Thank you also to Ms. Sherlyn Joy Isip for the blueberries testing that can give us a method or an effective histologic stain. To Mr. Sherwin Toriano, who leads this forum, thank you very much for your hard work. The insect vectors research also is a very good information that can prevent us for any infections. And the last but not the least, to Mr. Clive Ivan Mercado. Your research is very informative and your discovery of the Klebsiella infections can help us to prevent disease in the future. Collectively, all these results of our researchers and experiments today have led a significant improvement to our healthcare and transformation to the public health. My dear medical technologists, let me personally thank you for your presence here. Cannot imagine how in our life would be if we don't have that expertise of our medical technologies. We in PWU wishes that you continue all these endeavor and initiatives for the improvement towards the medical care of our Filipino people and most especially to the whole world. Congratulations to all of you. A round of applause to all the whole School of Medical Technologies. Good evening to everyone.
Thank you, Dr. Zernosa. We hope to hear from you again on our future activities. For further announcement, a link to an evaluation form shall be posted in the official MedTech FB page, and only those who have completed the evaluation at the end of this webinar are eligible to receive the certificate of participation. The certificates will be emailed on or before November 30, 2020, and please use your PWU email.
As the program comes to a close, we thank everyone for participating in today's online event. I've really learned much. How about you, Rania?
Me too. Research seems to be a very daunting subject for all of us, but with today's event, I am very enlightened and looking forward to explore the field of research.
We hope the rest of participants had a productive afternoon too. Again, I am Paulina de Guzman.
And I am Rania Maulana, your Masters of Ceremony for today. Thank you. Before we go, let us honor our alma mater and be proud of her legacy, the PWU hymn.